Thank you everyone for joining us this morning for this presentation. And myself, Dr. David Liam, and Dr. Dibakar Signal have been working on this project over the last few months. And the name of the project is Phrase Mining of Textual Data to Analyze Extracellular Matrix Protein Patterns Across Cardiovascular Disease. So I'll start off by giving a little bit of background information about the motivations of our study and by explaining how the accumulation and explaining how knowledge has accumulated in the form of textual data. So mounting information on cardiovascular disease in the form of textual data has accumulated. PubMed currently boasts more than 2.2 million CVD-related articles. Furthermore, over 100,000 ECM-related publications are accessible via PubMed. This rapid growth of biomedical literature and accumulated textual data is unmatched by the capacity of free text analytical tools to annotate and retrieve relevant biomedical insights from these vast amounts of data. As fundamental components of data science and bioinformatics, however, text mining and machine learning have emerged as powerful technologies to reveal meaningful relationships from unstructured textual data. This is because text mining is highly accurate, consistent, efficient, and can increase the functional value of unstructured data sets. With regards to accuracy, text mining algorithms can be designed to pinpoint relevant information, such as protein disease associations, minimizing individual bias. When a human completes the task of parsing through a report and extracting relevant information, individual biases are difficult to overcome. Furthermore, text mining algorithms provide reproducible results. Whereas when two humans manually mine a document, the information extracted may vary based on their individual biases. Additionally, text mining is highly efficient compared to manual methods of textual data extraction. Analysis of large volumes of information occurs within a reasonable time frame, such as 50,000 words per second or even 200 abstracts per second. Additionally, text mining transforms unstructured textual data into structured data with interconnected relationships. This highly increases the functional value of data sets. The aims of our study were to conduct a phrase mining and machine learning analysis of 1.1 million CVD-related abstracts across 709 ECM-related proteins. Our goal was to reveal hidden and unappreciated relationships between these 709 proteins and 6 CVDs to inform the underlying molecular pathogeneses of these CVDs. We aimed to apply this text mining and machine learning platform to analyze vast quantities of biomedical textual data to facilitate future phenotyping of these 6 diseases and as well as clinical decision making. Now I'm going to go over a more general workflow diagram of our study. So it begins with the collection of these 1,099,254 CVD-related abstracts. These are considered our unstructured textual data corpus. We then text mined these CVD abstracts using a variety of phrase mining algorithms. Our abstracts all were focused on six major heart diseases, namely cerebrovascular accidents, valvular heart disease, arrhythmias, ischemic heart disease, cardiomyopathies, and congenital heart disease. We aimed to define the relationships of 709 ECM proteins to each of these 6 CVDs. And based on machine learning, statistical, and pathway analyses of these raw data, we were able to reveal previously underappreciated relationships of the ECM to these 6 CVDs. So I'll start off by providing a little bit of background information on the extracellular matrix. The extracellular matrix is a non-cellular component surrounding all cells present within all tissues and organs, consisting of water, proteins, and polysaccharides. It provides physical support for cellular constituents, but also plays key roles in cell survival and function. It participates in signal transduction, that is, it carries signals from outside of the cell to inside to mediate gene transcription, And it also generates the mechanical properties of the organ, such as strength and elasticity. And it also mediates the protection of cellular constituents by maintaining extracellular homeostasis and water retention. We can observe in these diagrams how different components of the extracellular matrix integrate to provide structural support for cellular constituents. So this is a more specific workflow corresponding to our study. So it begins with our with the 6 CVDs that we chose and the associated mesh descriptors, and how we applied these to all PubMed research abstracts published between 1995 and 2016 to obtain our textual data corpus of 1 million CVD-related abstracts. And I'm going to go into a little bit more detail about these mesh descriptors in the next slide. In addition, we assembled a data set of 709 ECM-related proteins based on information available on UniProt and MatrixDB. We then integrated this ECM protein-related data set as well as our CVD-related abstracts and employed the novel phrase mining algorithm known as case-OLAT, or the context-aware semantic online analytical processing algorithm, to calculate a quantifiable relationship between each protein disease pair. This is based on 3 distinct criteria within case-OLAP: integrity, distinctiveness, and popularity. The final case-OLAP score is the product of these 3 quantities. And I'll be going into a little bit more detail about these equations and these quantities in future slides. Based on this raw data, we conducted various analytical methods to analyze ECM protein patterns and reveal underappreciated insights. So with regards to textual data corpus assembly, we extracted and screened 1 million CVD-related abstracts based on mesh descriptors. We assigned each of the 6 CVDs to 1 or 2 parent mesh descriptors. For cerebrovascular accidents, we use the parent mesh descriptor cerebrovascular disorders. For ischemic heart disease, myocardial ischemia. And for cardiomyopathies, we used 2 parent mesh descriptors, namely cardiomyopathies and heart failure. For arrhythmias, we used arrhythmias cardiac, and for valve disease, we used heart valve diseases, and for congenital heart disease, we used heart defects congenital. These parent mesh descriptors are linked to numerous mesh trees, and I'm going to go, and here is an example of how a mesh tree looks. And so we can observe that there are more general mesh terms, such as cardiovascular diseases, which are in a structured hierarchy with more specific disease terminologies. And so we refer to a term like arrhythmias cardiac as our parent mesh term, but we retrieved the articles associated with all of these descendant mesh terms, which describe more specific disorders. Another example of our parent mesh term can be seen at the bottom, cardiomyopathies. And now I'm going to go into a little bit more detail about our human ECM protein dataset assembly. We integrated 2 datasets from UniProt and MatrixDB to come up with our final dataset of 709 ECM-related proteins. Our criteria for choosing our dataset from UniProt was that experimental evidence or sequence similarity needed to confirm the presence of these ECM proteins in the extracellular matrix. And furthermore, all of our UniProt proteins were reviewed by UniProt curators to ensure legitimacy. Our MatrixDB dataset consisted of 660 human proteins curated using information extracted from the Matrixome project, UniProt, and gene ontology. The Matrixome project contains both core ECM proteins and ECM-affiliated proteins. Excluding proteins overlapping between these 2 datasets, our final dataset consisted of 709 human ECM proteins. In addition, when we parsed through our textual data corpus, we included the synonyms, abbreviations, and other known names of these ECM-related proteins. and all of this information was extracted from UniProt. And now I'm going to go into a little bit more detail on the case-OLAP algorithm. So the three criteria behind the case-OLAP algorithm are integrity, distinctiveness, and popularity. In these equations, we can observe that P in int PC stands for phrase, or in our situation, protein, whereas C refers to cell, and a cell is the subset of documents describing one of the 6 CBDs. And so with regards to integrity, our protein names were acquired from UniProt. Therefore, case-OLAP was not used to evaluate the integrity of protein names, and rather, since each of these protein names stand as meaningful semantic units, each protein received an integrity score of 1.0, which is the maximum score. Distinctiveness is a more subtle quality to define. Distinctiveness describes how a protein measures how exclusive a protein's relationship is to a particular CBD compared to the remaining 5 CBDs. The equation of distinctiveness first depends on this quantity, rel-PC, which is the relevance score of a protein within a subset of documents regarding one CBD. This score, rel-PC, is determined by comparing the occurrence of a protein name in a subset of one CBD documents compared to the remaining 5 CBDs. The final distinctiveness score was measured using this relevance score, and it was normalized to lie between 0 and 1. Finally, popularity measures the prevalence of a protein within a specific CBD. Popularity is only calculated based on the statistics from one cell of documents, that is, one subset of abstracts corresponding to cardiovascular disease. Popularity is a logarithmic function. Therefore, a rarely mentioned protein gets a very low score, while an increase in protein name frequency has a diminishing return. Again, we can observe that TFPC, the quantity, refers to the frequency of that protein name, where count PC is the summed frequency of all phrases in cell C. And then, like I mentioned before, the final case-OLAP score is the product of integrity, popularity, and distinctiveness. And this score is how we define relationships between the ECM proteins and the 6 CBDs. So our first step was to understand the strength of ECM protein disease relationships. And in this chart, we've displayed how different proteins are associated with the different diseases, and we've quantified the strength of their relationship. We can observe that a darker color corresponds to a stronger relationship. And see, the highest scoring protein out of all of the proteins was fibrillin 1. And fibrillin 1 was implicated in congenital heart disease, and it's a glycoprotein secreted into the ECM by fibroblasts that are essential for the formation of elastic fibers found in connective tissue. Deficiencies in fibrillin 1 and fibrillin 2 lead to Marfan syndrome and Beal syndrome, respectively. Other notable proteins in this diagram are nestin, which was found implicated in CVA, acetylcholinesterase, which was found implicated in arrhythmia, as well as apolipoprotein A1, apolipoprotein C3, plasminogen activator inhibitor 1, and lipoprotein lipase in ischemic heart disease. These are all known to play major roles in lipid metabolism and thrombosis and are risk factors for atherosclerosis. We can observe that ECM proteins display variable relationships to the 6 CBDs. The 6 CBDs do not have the same profile of ECM protein associations. And we explore these relationships further in the next diagrams. So this first plot on the left is a count plot, which counts how many proteins are shared between two diseases. We can observe that ischemic heart disease received the greatest number of ECM-related proteins, 211. And we can also see that the greatest overlap between two diseases and shared ECM proteins was seen in IHD with CVA, 147, and IHD with CM, 146. This may indicate a common molecular pathogenesis behind the roles of the ECM within these diseases. Now I'll move on to discuss our PCA analysis of these protein-disease relationships. PCA stands for Principal Component Analysis, and this is performed by analyzing 258 proteins that received associations by the Case-Olaf algorithm to the 6 cardiovascular diseases, and we basically produced a scatter plot of their associations by taking the two principal components of maximum variance. Each of the principal components is displayed in the figure below. We can observe that in principal component 2, there is a negative feature weight of ischemic heart disease, and that is why the ischemic heart disease vector is projected on the lower panel within this chart. We can also observe that the valvular disease, arrhythmia, and CM vectors are all projected closely together, while CHD and IHD remain projected further apart. This indicates similarities in the overall scoring patterns for cardiomyopathy, valve disease, cerebrovascular accidents, and arrhythmia, while ischemic heart disease and congenital heart disease have divergent scoring patterns. This indicates common molecular pathogenesis surrounding valve disease, arrhythmia, and cardiomyopathy, whereas congenital heart disease and ischemic heart disease may proceed. The roles of ECM in congenital heart disease and ischemic heart disease may be very disparate. We also aim to understand which ECM proteins play a unique role in the 6 heart diseases and which ECM proteins share roles with all 6 of the cardiovascular diseases. First, I'll go into more detail about the ECM proteins that were uniquely implicated in each of the 6 CVDs. What this means is that based on our Case-Olaf scoring mechanism, each of these proteins received a score only for one single disease, meaning that in arrhythmia, only one protein was found to uniquely score for arrhythmia, indicating that this protein has only been shown to be associated with arrhythmia in textual data. And so I'm going to go into a little bit more detail about the unique protein associations, protein disease associations in this study. We can observe in CVA that many proteins, that there were about 10 or 11 proteins found to uniquely score in CVA. This, excuse me, we found that these 12 proteins found to uniquely score in CVA predominantly play roles in signaling, nervous system development, and structural integrity. Pathways specific to nervous system development only appeared in analysis of these unique CVA proteins and not in any of the other 5 diseases. In cardiomyopathy, we noticed that the highest scoring proteins were ADP and ATP translocase proteins 2 and 3. These proteins were shown to have the greatest unique associations to CM. These 2 proteins catalyze the exchange of cytoplasmic ADP with mitochondrial ATP across the mitochondrial inner membrane, and the loss of these proteins has been shown to result in cardiomyopathy and myopathy. In congenital heart disease, we observed that there were about 5 proteins found to be uniquely associated with congenital heart disease. There were 2 microfibrillar proteins, and which are important constituents in the cell cytoplasm and the cell membrane, and they mainly consist of glycoproteins and cellulose. Many of these other proteins play a role in elastic fiber formation, and elastic fibers are connective fibers fundamental to heart valves in the cardiac ventricles, and thus they play a critical role in congenital heart disease. In IHD, we observed, in ischemic heart disease, we observed that the group was dominated by proteases involved in ECM remodeling, such as the A disintegrin and metalloproteinase with thrombospondin motifs family, as well as the matrix metalloproteinase families. We also found several proteoglycans and glycoproteins found to be uniquely associated with ischemic heart disease. Only one protein, nitrogen 2, was uniquely associated with arrhythmia. Nitrogen 2 is an ECM glycoprotein part of the basal lamina, and it likely plays a role in cell extracellular matrix interactions. In valve disease, we found 2 proteins with unique associations to the cardiovascular diseases. These are glypican 6 and 4. Glypican proteins are cell surface proteoglycans that bear the glycosaminoglycan heparin sulfate. They are known to be receptors for fibrovascular factor and various extracellular matrix proteins and proteases. Now I'd like to go into
Sanjana,
about some of the ECM
Sanjana, can you hear me?
Associations across all 6 CVDs. We found 82 proteins implicated with associations to all of the 6 CVDs. Based on a pathway analysis of these 82 proteins, we were able to, we revealed 2 notable pathways that have been underappreciated with regards to the role of the ECM in cardiovascular disease. And I'll be going into more detail about these 2 pathways in future slides. I'm sorry, Dr. Pink. Was there something you said?
I just noticed a typo. I wrote it in the box. So the valve disease, it's not 2 proteins. It's 2 isoforms of the same protein. We can correct it in the manuscript, no worries. I wrote a note, so I think David, everyone saw it too. Don't worry, please continue doing so well.
Okay, my apologies. I was muted for a second. But we performed a pathway analysis of the 82 proteins that shared ECM protein disease associations. When we performed this analysis, we found over 100 pathways that were found to be significant. We decided to further analyze the top 20 most significant pathways. And what we did is we quantified the relationship of each of these pathways to the 6 CVDs based on the proteins implicated in each of those pathways. We can observe that we confirmed existing pathways such as extracellular matrix organization, which is of course known to play an important role within the extracellular matrix and in cardiovascular processes such as remodeling. And we normalized our chart by this high score of 1, which extracellular matrix organization was found to be highly associated or played a significant role in ischemic heart disease. while we were able to confirm existing pathways, we were also able to highlight previously underappreciated pathways with regards to the role of the ECM in cardiovascular disease. These are interleukin 4 and 13 signaling and regulation of insulin-like growth factor transport and uptake by insulin-like growth factor binding proteins. Both of these pathways were found to receive moderate to high scores across all 6 CVDs. And I'm going to go into a little bit more detail about them right now. So, like I said, all six CVDs exhibited moderate to strong relationships to the pathway regulation of insulin-like growth factor and uptake by insulin-like growth factor binding proteins. Insulin-like growth factor possesses insulin-like metabolic actions and growth factor-like actions during cell proliferation and differentiation. This IGF promotes cell proliferation and it inhibits apoptosis. And this overarching effect provides a metabolic effect as well. It provides a signal to cells that adequate nutrition is available, and thus cells avoid apoptosis, enhance cellular protein synthesis, and enable cells to undergo hypertrophy in response to an appropriate stimulus. Furthermore, we have learned that the insulin-like growth factor regulates contractility, metabolism, hypertrophy, senescence, and apoptosis within the heart. Furthermore, IGF-1 has strong implications in metabolic syndrome. Metabolic syndrome is a cluster of risk factors such as obesity, dyslipidemia, hypertension, and insulin resistance. The collection of risk factors known as metabolic syndrome is a major risk factor for all of the 6 CVDs. And the mechanisms underlying the actions of IGF-1 in metabolic syndrome relies on ECM proteins, including fibronectins, laminins, and integrins. Fibronectin is a glycoprotein within the ECM that binds other ECM components and plays a structural role. Laminin is associated with collagen in ECM and it interacts with integrin proteins. Integrin proteins are transmembrane receptors that facilitate cell ECM adhesion. These 3 different types of ECM proteins have been implicated in insulin-like growth factor actions, yet the connection between these proteins, the insulin-like growth factor axis, and cardiovascular disease has been previously underappreciated, yet was brought to light by our study. I'm also going to go into more detail about interleukin 4 and interleukin 13 signaling. As I previously mentioned, all of the 6 CVDs exhibited moderate to strong relationships to this pathway. Interleukin 4 and 13 are highly identical cytokines, and their dysfunction is known to underlie the pathophysiology behind allergies and inflammatory disease. However, relatively little is understood regarding the role of interleukin 4 and 13 in cardiovascular events. Furthermore, relatively little is reported about the role of interleukin 4 and 13 in cardiovascular events. However, in 2017, a study was published that linked the downregulation of interleukin 4 and 13 receptors with heart failure. The downregulation of these receptors was further associated with collagen deposition during pressure overload conditions. Collagen deposition is an ECM-mediated process. This study published a new role between interleukin 4 and 13 and cardiovascular disease in 2017. But our study, which analyzed abstracts published between 1995 and 2016, was able to identify a link between these signaling pathways before this relationship was discovered. This highlights the ability of our analytical method to connect dots between protein disease associations and provide conclusions that may be otherwise underappreciated or hidden within textual data. Now, this is our last analysis presented in this study. And so here we performed hierarchical clustering analysis, where we treat each protein as a 6-dimensional vector based on the 6 cardiovascular disease scores received. And we plot the proteins based on an equation known as the standardized Euclidean distance metric. This enables us to cluster proteins with dominant associations in a particular CVD. And we can observe that there are about 6 major clusters in this within this presentation. The first on the upper right, and then over here, and 3 lining to the bottom right corner. We have highlighted dark areas of each of the 6 clusters with a red square. This denotes proteins that were uniquely scored within that cardiovascular disease, and this can be observed by the color key to the right of the hierarchical clustering analysis. As you can see, this red rectangle, the 2 red rectangles line up, and we can observe that the proteins in the dark center portion of the cluster all uniquely scored for cardiomyopathy. The same occurred in 4 of the 6 clusters. This further highlights the role of these unique ECM proteins within these 6 cardiovascular diseases. We further characterize these clusters in this table. Here we've presented the top 5 proteins in each cluster, and these top 5 proteins were determined based on the strength of the associations they displayed to each of the 6 CVDs. We quantified the strength of these associations, of course, by using the Case-Olaf score. We then have determined notable biological pathways in each of the protein clusters based on a reactome-empowered analysis. We've highlighted one interesting pathway within arrhythmia, notably virus assembly and release. Virus assembly and release was found implicated in arrhythmia by our study. And upon further literature review, we learned that many viral infections, such as Coxsackie adenovirus, are known to result in myocarditis, pericarditis, or endocarditis. These viral infections impact tight junctions among cardiac cells, leading to the development of various arrhythmias. Furthermore, the Coxsackie adenovirus receptor, CAR, regulates cardiac conduction and remodeling. CAR is also essential to virus uptake and assembly. Lastly, several ECM proteins, such as fibronectin and laminin, have been shown to interact with CAR and to enhance the barrier function of tight junctions. Taken together, this evidence supports a functional link between the relevant ECM proteins and viral receptors. Importantly, by quantifying ECM protein disease associations across all 6 CVDs, our analysis was able to pinpoint the unique impact of the virus assembly and release pathway in arrhythmia, but not in the other 5 CVDs. This highlights the precision of our analytical methods. So in summary, by connecting data points of protein associations, a combination of text mining algorithms, machine learning analyses, and statistical tools efficiently provided novel biomedical insights among 709 ECM proteins and 6 cardiovascular diseases. Reactome-empowered pathway analyses of ECM protein associations with all 6 CVDs revealed underappreciated associations of the ECM and cardiac pathophysiology, namely with regards to insulin-like growth factor regulation and interleukin 4 and 13 signaling. Hierarchical clustering analysis aggregated proteins according to the dominant role they played in a targeted CVD, and as such, the relationship between ECM proteins and viral pathways in arrhythmia was highlighted. Taken together, these insights that we acquired support the role of text mining to uncover novel associations in biomedical literature and eventually complement other biomedical technologies to further facilitate phenotyping and clinical outcomes. We are now employing Case-Olaf to study the relationships between 8,000 plus heart-related proteins with the 6 categories of CVD. finally, we'd like to thank Professor Jiawei Han and his team at the University of Illinois at Urbana-Champaign. And I'd also like to thank Dr. Ping, Dr. David Lim, Dr. Sigdal, Vincent, and Howard for all of their hard work for putting together this presentation. Thank you all so much, and please let me know if you have any questions.
Does anyone have questions for Sanjana?
I have oh, Andrew, go ahead.
Thanks. Just a very quick question. Very nice work, very interesting. So you've clearly restricted to ECM proteins based off of sort of a known knowledge and previous hypotheses. But I guess I wonder, are there any, what are the biggest challenges with extending it to essentially any concepts recognized in the text, regardless of whether, other classes of proteins or even other classes of entities?
That's a great question. We chose to restrict our analyses to ECM proteins in this study because we wanted to work with a manageable data set with a large body of known information. Of course, Kisolov is definitely capable of extracting relevant phrases and utilizing different phrases that we could have input it with. But in this study, we wanted to really validate this algorithm and its application in biomedical literature. And so we chose to focus on a smaller subset of proteins.
That was excellent. So, Sanjana, I am so proud of you. Andrew, I'm adding to what Sanjana has said. Everything she said is right on the target. So you probably aware there are quite a few projects going on using or adapting the application of this Kisolov platform Professor Jiawei Han has built. The ECM proteins has been one of the parallel projects, but it kind of got moved forward because the special issue call on ECM proteins. And this, with 700 proteins, it is much more manageable comparing it to looking at these relationships and disease and phenotyping them to a much bigger data sets. What we've also done in this particular study, and Sanjana had gone into details explaining how MASH descriptors are applied, and those are something that goes through very careful considerations of what are the parenting MASH descriptors and what are the relevant descendant or daughter descriptors, and if we should be using only MASH terms or should we do combination of MASH and ICD-10. And so it took us some time and effort to work out these technical details until we felt comfortable that the descriptors are collecting the information we aim to target. And so it's quite a bit of effort there. And so one of the parallel projects is the clinical case study projects with Harry and Anders, and as well as John, he's a cardiologist, with David. We've done the combination of the MASH descriptors and the ICD-10 codes. And so those were things we didn't encounter that we would have some struggle, but we did, because Andrew, you probably know, it's limited, but there are overlaps where the daughter MASH terms intrude into different space of the parenting MASH terms. So these are now worked out. I feel very comfortable, I'm happy. And the study Sanjana has just presented, we're now in minor revision, this manuscript. So we anticipate it to get accepted this round in. and so we can focus on the other big projects. The algorithm is extremely powerful, Andrew. At this moment, we're looking at, for the ECM project, we're looking at only one dimension, that is protein and disease phenotypes. And in other projects, we begin to explore second and third dimension, looking at biomarkers, therapeutic device, and protocols. So I'm very excited about this project and the direction we're going into.
Great, thank you both.
So I want to congratulate Sanjana. She's been just amazingly smart and brilliant in working with both David and Devaka on this project. And in the meantime, she did something like 17 med school interviews. Has been accepted by at least 3 medical schools, Michigan, Case Western, and we just been thrilled to have her as part of our team and made amazing contributions, very, very smart, brilliant contributions to this project. Thank you, Sanjana.
Thank you very much, Dr. Ping. I really appreciate the opportunity.